Welcome friends to a new session on application of Perl to bioinformatics. Here we are discussing the remaining part of mutations and randomization. Mutations and randomization part 2. Now we have a general idea of how a random DNA can be generated using computer program codes. Random data is usually generated for test purpose. The random DNA can also be used to study the organization of actual DNA from an organism. There are two different approaches for program designing, such as top-down and bottom-up. A top-down approach is essentially the breaking down of a system to gain insight into its compositional subsystems. In this approach, an overview of the system is formulated specifying the subsystems. Each subsystem is then refined in detail, and if necessary, some additional subsystems are defined until the entire specification is reduced to base elements. Hence, the top-down approach starts with the big picture and breaks down from there into smaller segments. A bottom-up approach is based on incoming data from the environment to form a perception. In a bottom-up approach, the individual base elements of the system are first specified in great detail. These elements are then linked together to form larger subsystems, which then in turn are linked sometimes in many levels until a complete top-level system is formed. Here, the strategy is that the beginning are small but eventually grow in complexity and completeness. Genes control the majority of functions of an organism. Mutations can lead to changes in the structure of an encoded protein or to decrease or complete loss in its expression. Mutations can cause damage to a cell or organism because a change in the DNA sequence affects all copies of the encoded protein. A mutation may involve a change in a single base pair called a point mutation, or a deletion of a few base pairs generally affect the function of a single gene. Some other mutations involve large-scale changes in chromosome structure and can affect the functioning of numerous genes, which results in major consequences in the physical appearance. Many common human diseases are due to the mutations in single genes. Randomization is a computer technique that is very common in everyday programs, especially in cryptography. For example, if you want to generate a hard-to-guess password, you are using randomization. Using randomization, it is possible to simulate and inspect the mechanism of mutations in DNA and their effect upon biological activity of their associated proteins. A powerful tool for studying systems and predicting what they will do is simulation, and randomization allows to better simulate the ordered chaos of a biological system. Simulating mutations with computer programs can aid in the study of evolution, disease, and basic cellular processes such as division and DNA repair mechanisms. To model evolution, mutation, and disease, randomization is a powerful and easy to use programming skill.